Hi everyone and welcome to Miss Estruch Biology. In this video we're going through the whole of topic 7. If you are new here, then I'm Miss Estruch and I've been teaching biology for over 10 years as well as study skills and revision skills. So if you need support with any of that, then make sure you click to subscribe so you don't miss out on any of my latest videos. I also have lots of resources on my website to help you. I've got my free exam question bundles, but also if you need that extra boost in helping you to learn the content, then check out my A-level notes. In these, I go through all of the key marking points that come up on AQA papers using my knowledge of over 10 years of teaching AQA. I also have lots of essay links to help you with that too. So if you do want that extra help to boost your grade up, then click the link below. Now, don't just take my word for it. I've had so many students saying how helpful they found my notes. They've said they've managed to go up so many grades just from using the notes alongside these videos. So if you are needing that extra push before the exams, then there they are. So the first thing with inheritance is there are a lot of key terms that come up quite often as a 1-mark definition just to start off an inheritance question. So make sure you know all of these definitions as they've all come up at some point in the past. So genotype is the genetic constitution of an organism. Phenotype is the expression of the genes, but also the interaction with the environment, so don't forget that bit. Homozygous is when you have a pair of homologous chromosomes carrying the same alleles for a single gene, whereas heterozygous is when you've got the homologous chromosomes carrying 2 different alleles for a single gene. Recessive allele is only expressed if there's no dominant allele present, whereas the dominant will always be expressed. Codominant is when both alleles are equally dominant and expressed in the phenotype. Multiple alleles is when you have more than 2 alleles for a single gene. Sex linkage is a gene whose locus is on the X chromosome. Genes are located on the same chromosome, not the sex chromosomes though, for autosomal linkage. Epistasis is when one gene modifies or masks the expression of a different gene at a locus. And then monohybrid and dihybrid is referring to types of genetic crosses. So monohybrid is when you're looking at the inheritance of just one gene. Dihybrid is when you're looking at the inheritance of 2 genes at a time. And we're going to go through the types of inheritance in more detail and have a look at examples of genetic crosses. Now, a big part of the inheritance topic is being able to determine the outcome of genetic crosses. So to help you with that all in one place, I've got you this genetic coding table. So for the different types of inheritance, this is how you would represent it in a genetic cross. A monohybrid is what you would have done at GCSE. So you pick a letter to represent the gene, capital is the dominant allele, lowercase is the recessive. For codominant, because they're both dominant, you can't go for a capital and a lowercase. So instead, the base letter is representing the gene and the superscript letter is representing the allele. So for example, blood group is IB for blood group B, IA for blood group A, but if you have this here, IBA, that would mean that it's someone that's blood group AB because both dominant alleles will be expressed. Multiple alleles, again, you can't use the upper and lower case because you have more than 2 alleles, and you might have this linked with the idea of codominance as well, as we see in the example of blood groups. The allele that codes for blood group B is dominant, the allele that codes for blood group A is dominant, but there are multiple alleles because there is a third one for blood group O, but that is recessive. For sex linkage, you have to represent this by showing first of all the X and Y chromosome, and in sex linkage the gene is only found on the X chromosome, so you'll only be drawing the allele on the X chromosome, not on the Y chromosome, but we still stick to capital for dominant, lowercase for recessive. Autosomal linkage is always linked to dihybrid because it's looking at the inheritance of 2 genes found on the same chromosome. So we can still use the capital and lowercase, but we'd have 2 different letters representing the 2 different genes. Epistasis would be the same idea as well, because they often don't link it to any of the other types of inheritance because epistasis is complicated enough. So now for the examples. So we're starting off with a simple monohybrid example where we're looking at the inheritance of cystic fibrosis, which is caused by a recessive allele. And we're told in this question that we have two parents that are heterozygous, so we can see that represented here. And we're being asked what is the probability they will have a girl with cystic fibrosis. Now don't be thrown, this is not a sex-linked gene, but they still want you to work out the probability of, first of all, whether the child will have cystic fibrosis. And we can see here we have 25% probability that a child would have cystic fibrosis. But because they want you to say the probability of a girl having cystic fibrosis, there's 50% probability they'd have a girl. So we'd then do 25% times 50%, or half of it basically. So there's 12.5% probability that they'll have a child who has cystic fibrosis who is also a girl. Now if we have a look at a codominant example, cows can be red, white or roan in colour, and this is roan here where it's blotchy with both colours expressed. And in this example we have two roan cows reproducing, and they want to know what's the probability they'll produce red offspring. So two roan cows, because this is codominance, both the red and the white are dominant. That's how it's possible to get roan where they're both expressed. So here is how we'd represent it for the two parents. And then if we do our Punnett square, we can then see we'd have one roan, one red, actually a second roan down here, and one white. So that would mean we have 25% probability of getting a red cow from those parents. Looking at the inheritance of blood groups is an example of multiple alleles and codominance. So here we have the phenotypes that are possible of the different blood groups. Now you don't need to know this. If they wanted you to work out anything to do with blood groups, they would have to give you some information so you could work it out. It's not on the spec, you have to know the blood groups. It's just a really common example to test your understanding of codominance and multiple alleles. So blood group A, you could either have two copies of the dominant A, or one copy of the dominant A and one recessive O allele. Same idea for blood group B, but with the B alleles. To be blood group AB, this is our codominance where we have one copy of both of those dominant alleles, so both are expressed. Blood group O is recessive, so it's only possible if you have two copies of IO, which is our recessive allele. So if we have a look at an example, we have parents with blood group AB and O reproduce, and we're told that A and B are codominant, O is recessive, because as I said, you're not expected to know that. It would be application of your knowledge. And they want to know what's the probability they'll have an offspring with blood group A. So the first thing is you have to state the parental genotypes. Then we can work out the gametes, which I've put here on the outside of the Punnett square. Then we have the offspring genotype and the offspring phenotype. So we can see the probability of having blood group A is 50%, because we have two individuals down here that have both got a genotype that codes for blood group A. So now our example of sex linkage, it's looking at colour blindness, which again you don't need to know, it's an application of your knowledge, and you're told that colour blindness is caused by a recessive allele found only on the X chromosome. If a non-colourblind male reproduces with a female carrier of the allele, what is the probability their children will be blind? So first of all, we need to work out the parents' genotypes. So the mother is a carrier, so that means they'll have one dominant allele and one of the recessive alleles that causes colour blindness. And then the male, XY, they do not have colour blindness, so that means they have to have just the dominant allele on their X chromosome. So here we have our gametes. We then have the offspring's genotypes and phenotypes. So the probability that they would have a child that is colourblind is 25%. So epistasis, we said, is when one gene influences or affects, masks the expression of another. So some examples are the coat colour in mice, or in Labradors, fruit colour of vegetables as well, such as squash. And we'll have a look at the Labrador example. So gene 1 controls whether pigment will or will not be expressed, and the dominant allele codes for pigment will be produced, whereas the recessive codes for no pigment, and therefore the dog won't be producing any of the darker pigments and it'll be that yellow colour. Gene 2 controls what colour the pigment will be if it is expressed. So dominant is black and recessive is brown. So that is how you get the 3 different colours in Labradors. So we have a look at an example. We're going to cross these 2 parents. So the first thing, we've been given the genotypes, but we have to work out the phenotypes. Now both of them have a dominant E, which means pigment can be produced, and they have a dominant B, which means they'll be black. After that, it's working out all the possible gametes that we could have. Then we'd need to do a Punnett square to work out all the possible genotypes of the offspring, and then the phenotypes of the offspring, which I've done for you just here. So we can see we end up with 4 yellow dogs because they have homozygous recessive for that E gene. So even though they might code for black and brown, we won't see that colour because it's been masked by the first gene. Now dihybrid crosses are when we have 2 genes considered at the same time in a Punnett square. So a common example is Mendel's peas. So we've got round and yellow, wrinkled and green as our 2 different traits, the shape versus the colour. And in all of the genetic crosses that we've gone through, it's these 6 features you have to have for all of the marks. We need the parental phenotype, genotype, the gametes, offspring genotype, and then you match the offspring phenotype to the genotypes, and finally give the proportions. So if we have a look at this example, with our round and yellow, round is dominant and yellow is dominant, wrinkled and green are recessive. And in this example, although we've got the 2 dominant traits, they are homozygous dominant. And they would have to tell you that in the question, or they would have to give you information so you could work it out. So then we'd have a look at our gametes, and each gamete should have 1 copy of each gene. So you need 1 of the letter R's for the shape, 1 of the letter Y's for the colour. Now in this instance, that would mean that all of the offspring would be heterozygous, and you'll always see this pattern. When a homozygous dominant parent is reproduced with a homozygous recessive parent, you will always get 100% of the offspring being heterozygous and therefore showing the dominant trait for both of those genes. Now if we cross the F1 generation though, where both parents are heterozygous, there are now 4 possible different gametes for both parents. And this would be where we get these large Punnett squares where we put every possible gamete for both parents in the outside box. Then we work out the offspring genotypes, and then we work out all of those phenotypes, and I tend to put it in the box so you are then definitely getting that mark of matching the phenotype to the genotype. So our ratio here is 9:3:3:1. And if you have a dihybrid cross where both parents are heterozygous for both genes, then it will always be 9:3:3:1, unless there is autosomal linkage or there is crossing over. So let's have a look then at crossing over and how this could affect the ratio. So crossing over results in new combinations of alleles in the gametes. So that means the predicted gametes that we had in our Punnett square may actually differ. So autosomal linkage is when 2 genes are located on the same chromosome, but it won't be the X or Y chromosome. So we can see here we have gene 1 is found on the same chromosome as gene 2. And this is showing us the homologous pair of that particular chromosome. So what this means is that the alleles for each gene are linked on the same chromosome. So they have to be inherited together, because in meiosis, when gametes are made, that whole chromosome will be pulled to create one particular gamete, and this chromosome will then be pulled to the other pole to create the other gametes. So that would mean you would have to have the 2 dominant alleles for the Y and R gene inherited together, and the 2 recessive alleles for the Y and R gene inherited together, as those would be the only 2 types of gametes that could be made. So if we go back to the dihybrid example where we said we have 2 parents that are both heterozygous, we said these were all the possible gametes that could be made. But if there was autosomal linkage, even though we have 2 heterozygous parents, Because the R and Y gene are linked on the same chromosome, and we can see we have the 2 dominants and the 2 recessive alleles on individual chromosomes, that actually means the only possible gamete would be one that has the dominant R and Y and one that has the recessive R and Y. So our possible genotypes of the offspring and the phenotypes are here, and we get a very different ratio, a ratio of 3 to 1, and that then does not fit the typical 9 to 3 to 3 to 1 ratio. So that is how autosomal linkage could prevent you from getting that ratio. But we also need to consider crossing over and autosomal linkage impact as well. So we've just talked through this example where, because of the autosomal linkage, we have a 3 to 1 ratio, but you could actually have this scenario here. Scientists actually observed the following results. So we have 4 different phenotypes, not just 2. And we can see we mainly had yellow and round, and the least common was the green and round. And this is only possible because crossing over has occurred to create new combinations of gametes. So not just these 2 gametes that we said would be possible, new combinations have been made, so there's now actually more gametes. So this is how it would have happened. We can see that we said we'd have the 2 recessive alleles linked and the 2 dominant alleles linked. If we then have crossing over, we can see that the recessive R and the dominant R cross over, that bit of the chromatid will break and exchange, and we now have 4 possible gametes instead. Now you also need to be able to see how chi-squared, the statistic, can link to inheritance. So chi-squared is used when you're investigating differences, but it's when your data is based on categories and frequencies. So if we go back to the very first example we had in this video, this is what we were looking at, the probability that 2 carriers would have a girl with cystic fibrosis. And we said we would expect the probability to be 12.5%. Now, chi-squared is the statistic used to investigate the differences between frequencies, but more specifically to determine whether there is a significant difference between the frequency we expect from our Punnett square and the frequency you actually observe. So let's go through an example. Corn can be purple or yellow and smooth or wrinkled. It's not actually just yellow like you see in the supermarkets, and this is determined by genetics. So using a Punnett square and by counting the actual number of each type of kernel, we can then use chi-squared to see, is there a difference between the expected frequency and the observed frequency? So does the frequency of the purple and yellow corn match the Mendelian genetics you would expect, which basically means the expected value you get from a Punnett square. So purple is dominant, yellow is recessive. And we've got this particular cross from 2 parents, and we would expect from this that we would get a 3 to 1 ratio of purple to yellow. A student then counted the actual number of each coloured kernel, and they recorded 21 purple and 13 yellow. So we can then use the chi-squared to see, do we have a difference between the expected ratio and the observed ratio? So we always state that there is no significant difference between the expected and the observed frequencies in the null hypothesis. And what we'd first have to do is convert the ratio, which is what we use in the Punnett square, into an actual expected frequency. So this is what was observed. So the total number of corn was 34. So if we have a 3 to 1 ratio is what we expect, the frequency that we would expect from that value would be 25.5 and 8.5. We would then do our observed minus expected. We square that, divide that by the expected, and we get our chi-squared value. In this example, the number of degrees of freedom is 1 because it's the number of categories minus 1. And we have 2 categories, purple or yellow. So the last thing you would need to do would be compare your chi-squared value to a critical value. And in this case, the calculated value of chi-squared we said was 3.176, and our critical value, we have 1 degree of freedom, so we need to look across at the row 1 and we go to 0.05, and that would be telling us that there'd be 5% probability the results are due to chance. We always compare at this point to be able to say if we have a significant difference or not. And in this case, we can see that our calculated is less than the critical value. So that means there's more than 5% probability the results or the difference between the observed and the expected is due to chance. So that means we have to accept the null hypothesis, which was that there will be no difference between the expected and the observed frequency. And what that means is the expected frequency that we calculated using our Punnett square was the same as the frequency we observed. Or in other words, it did follow the Mendelian genetics that we expected.
The Hardy-Weinberg principle, which is essentially a mathematical model which can be used to predict the allele frequencies within a population, which means how common one particular allele is within that population's gene pool. So just to review some of these key terms then, gene pool, population and allele frequency. So the gene pool is all the alleles of all the genes within a population at one time. Population is all the individuals of one species in one area at one time, and they can interbreed to make fertile offspring. And lastly, the allele frequency, that's the proportion of an allele within the gene pool. So these are key definitions to learn for AQA, but it's also to help get your head around the calculation, which we're going to go on to next.
The Hardy-Weinberg equation is this one here at the top, but you actually use both of these equations simultaneously. And the main thing you need to be confident with is, first of all, knowing those equations, but secondly, knowing what each component means. So a single letter by itself is an allele, and the P is representing the dominant allele. The Q is the recessive allele. These ones here in blue are the genotypes. So P squared is homozygous dominant genotype, meaning 2 copies of the dominant allele. 2pq would be heterozygous genotype because we've got 1 of each letter, and q squared is the frequency of the homozygous recessive genotype. So we use these 2 equations to be able to work out any one of those components using the data you're given. So in this example, we're told that cystic fibrosis is caused by a recessive allele, so that would be the letter Q. 0.02% of the population suffer from cystic fibrosis, and you then need to use that information to work out what proportion are carriers. Now, the first thing you should always do is identify which parts of the formula you have values for. So if we've been told that that percentage of the population are sufferers, that means that is our genotype Q squared, because they must have 2 copies of the recessive allele to be sufferers. So we've been told that Q squared is 0.02%, but we can't leave it as a percentage because in our Hardy-Weinberg equation, we say that the total equals 1, not 100. So it's not as a percentage, we need it as a decimal place. So we divide that by 100 to turn it into a decimal. Next then, we work out P and Q. So if we have Q squared, that means we can work out Q by square rooting that value. So there is our Q value. And if you have Q, we can then rearrange that second equation, making P the subject, and it would be 1 minus Q. So P is 0.986. And once you know what P and Q are, you can work out any other part of the equation. Now, they've said they want you to work out what proportion are carriers. And if they're carriers, that must mean that they are heterozygous in this example. So they're asking you to work out 2pq. So 2pq would be 2 times p, so 2 times 0.86, times q, which is 0.014, so 2pq is 0.03. And you need to look carefully to see if they specify your answer has to be given as a percentage or not. If it doesn't say anything, then you can leave it as 0.03. If it says a percentage, then times it by 100. So next we're considering variation. And individuals within a population of the same species may show a wide range in variation of phenotype. And this is due to both genetic differences but also environmental factors. The main source of genetic variation is mutations, but there will also be some genetic variation introduced by random fertilization of gametes and also in meiosis, crossing over and independent segregation. Now that variation is what leads to natural selection. Predation, disease, competition are all things that are going to be driving this natural selection. And as a result, Those organisms with the phenotypes providing the selective advantages are more likely to survive and therefore pass on their favourable alleles to the next generation. This changes the allele frequencies within the gene pool, because whichever allele gave the selective advantage will become more frequent. So that takes us on to this idea of types of selection. Now, in topic 4, you learn about stabilizing and directional. What we're going to be covering is just disruptive, because that's the only one that comes up in topic 7. And disruptive selection, which we can see up here, is when individuals which contain the alleles coding for the extreme traits are more likely to survive and pass on their alleles. So as a result, the allele frequency changes over many generations, and more individuals end up having those two extreme traits. And the middling trait is lost, because having that trait is not an advantage to survival, so it won't be passed on to the next generations. Now, that disruptive selection ultimately can result in speciation, which is the creation of a new species. And this occurs when one original population of the same species becomes reproductively isolated. And that isolation means that there are now two populations of the same species, but they will not breed together. So that can result in the accumulation of differences in their gene pools to the extent that those two populations would be unable to interbreed and make fertile offspring if they were then able to reproduce. So there has to be something that is preventing them from reproducing with individuals from those separated populations. And therefore, over many, many generations of those two populations accumulating different mutations, we end up with them having such different DNA that they cannot make fertile offspring. Now, that could be allopatric speciation or sympatric. So allopatric is when there is a geographical barrier, so that could be a new mountain range, for example, whereas sympatric is when there are reproductive mechanisms preventing it. So in allopatric speciation, a population can become geographically isolated over time. It could be a new mountain range or new bodies of water separating the land masses, and that separates the original population into two. So they are now unable to reproduce due to that geographical barrier. Both of those populations will be accumulating different beneficial mutations, and over many, many generations, that will then result in their DNA becoming so different that they cannot interbreed to create fertile offspring. And therefore we class them as two new species. Sympatric, we said, is when the populations become reproductively isolated, and this is due to differences in their behaviour. So they are not geographically isolated, they are in the same area, but they still won't reproduce. This could be because a random mutation has impacted the reproductive behaviour. So for example, it might cause individuals to perform a different courtship ritual, or maybe they now become fertile at different times of the year. So even if they did mate, they wouldn't become pregnant, and therefore they would not be mixing genes within that particular gene pool. Due to this, these individuals will not produce together, and there'll be no gene flow between the populations. Over time, these reproductively isolated populations, just like in the other example, accumulate different mutations to the extent that their DNA is so different they are no longer classed as the same species. Genetic drift is the change in the allele frequency within a population between generations, and there will always be genetic drift from one generation to the next, but continual, substantial genetic drift is what results in evolution. And the smaller a population is, the bigger the impacts allele frequency changes have proportionally, and this is why evolution occurs more rapidly in smaller populations.
For this part of topic 7, the populations, there's a lot of key terms that can come up as just 1 or 2 mark questions in the exam. So these would be great to turn into flashcards or just to test yourself. You can screenshot and take a copy of this table. So populations is a 2 mark definition, and your definition is it's a group of organisms of the same species living in the same area at the same time. That first part is 1 mark. The second mark is that they can interbreed to make fertile offspring. Habitat would just be 1 mark, so it's the part of an ecosystem in which particular organisms live. Community, so this is all populations or all the populations of different species in the same area at the same time. and it is essential that you stay in the same area at the same time. The ecosystem, so that is the community and the non-living components of the environment, so essentially the biotic and the abiotic factors. And you need to have an awareness that the ecosystems can range in size from being very small to very large, like global. Niches is often a 2-mark definition as well. So it's an organism's role within an ecosystem, including its position in the food web, but also its habitat. And it's normally split as it's an organism's role within an ecosystem, and then the second mark is also the habitat also. And each species occupies its own niche, and no two species can occupy the same. And if they do try, then that's when you get competition, and one species will eventually be outcompeted. Carrying capacity is the maximum population size that an ecosystem can support. And if you exceed that carrying capacity, there then won't be enough resources for all of the individuals, and they'll start to die, and that will then bring it back to the carrying capacity level. Abiotic factors are the non-living conditions of an ecosystem that affect the survival of individuals. And the biotic factors are the impacts of the interactions between organisms.
We have a look then at the abiotic factors and the biotic factors. Abiotic factors, as we said, are the non-living components of the ecosystem, and these can be factors like the temperature, oxygen and carbon dioxide concentration, light intensity, pH and soil conditions. Plants and animals are adapted to the abiotic factors within their ecosystem. These adaptations develop through the process of natural selection over many generations. The less harsh the abiotic factors, so for example if there's plenty of water and light, the larger the range of species and the larger the population size. So for example, in the desert, because it's very harsh conditions, you'll have a smaller range of species and the population size is smaller. Compared to down here where we can see there's plenty of light and water, we have a bigger range of species and there's more of each species. Biotic factors, we said, are the living components that are interacting together. So for example, we could be looking at competition, whether it's interspecific or intraspecific, and also the effect of predation. So our two types of competition. Interspecific competition is when members of different species are competing for the same resources that are in limited supply. Whereas intraspecific competition is when members of the same species are competing for the same resources and also a mate. And competition for a mate links to the courtship rituals. The predator-prey relationships you will see presented in graphs like this, which are called predator-prey graphs. And regardless of the species, the graph always follows a very similar pattern. The size of the predator and prey populations will both fluctuate. There will typically be more prey than predators because there has to be more prey to sustain the predators because they are eating them. And the size of the population change will always have a slight lag. So whatever happens to the prey, that will then happen to the predators shortly after. So the prey will increase first, then the predators will. The prey will then decrease because they're being eaten by the predators. But then because their population decreases, so will the predators. So if we think about sampling, first of all, why do we have to sample? Well, if you were asked to work out how many daisies there were in this field, you would have to do sampling rather than count every individual one. It's more time efficient and you're actually more likely to get an accurate answer because you'd probably miscount if you had to count all of those. But to make sure sampling is actually going to accurately represent that population size, you have to make sure that you are randomly sampling if it is a uniform area, and that's to eliminate bias. You also should have a large sample, so take at least 30 samples. Now I've got here a sampling summary just to help you see how it all links together. So if you have a slow moving or non-motile organism like plants, you would sample using a quadrat. If it was uniform distribution, you would do random sampling. If it wasn't evenly distributed, so for example a river in the middle of your field, then you would do a line transect. If you have a motile organism, meaning they can move, then you would need to use Mark-release-recapture. So let's have a look at these three types of sampling. So first of all, random sampling. This is when you'd have two tape measures pulled out at right angles to create a gridded area. You would then need to use a random number generator to get two coordinates, and those coordinates indicate where you would place your quadrats. Once you place your quadrat, you then collect the data, and you would then repeat this at least 30 times and calculate a mean. Now, if you didn't have an even distribution, we would use a line transect. So common examples of when you need a line transect are rocky shores or any kind of beach fronts, as well as maybe going across a path or a river. So for example, if we wanted to have a look at the impact of the river, you would put your line transect going across it. If you wanted to know the impact of the rocky shore, you would go from the rocky shore going further away into the land. You can either do a belt transect or an interrupted belt transect. A belt transect is when the quadrat is placed at every single position along the tape measure. An interrupted belt transect is when you place it every set distance. So it might be every 2 meters, every 5 meters, whatever seems appropriate for the area you are sampling. So for this method, you would place the tape measure at a right angle to the shoreline, just like we see here. You would then place your quadrat every 5 meters, or whatever position distance you're going to state. Collect the data that you can see in the quadrat, but you still have to repeat this multiple times. But you would be repeating this by placing another 30 transects along the beach at right angles to the shoreline. And you have to be really specific with how you word number 4. If you just said repeat 30 times, you won't get the mark. You have to describe the position. Now, mark-release-recapture you would use for moving animals. So first of all, we have the method here. An initial sample of the population in question is captured. These individuals are then marked and the number caught is recorded. And the mark has to be all weather resistant, so it doesn't come off, but it also has to cause no harm to the organism. The marked individuals are then released, but you have to leave them for long enough to evenly distribute across their habitats before you then take your second sample. And when you take your second sample, the total number captured in that sample is recorded, as well as how many you recaptured that had the marking on them. And the size of the population can then be calculated using that information. But as with all sampling, the more times you repeat it, the more reliable your results should be. So here's our calculation. To estimate the total population, you would do the number of organisms initially caught times by the number of organisms in the second sample, divide that by the number of organisms that you recaptured. So here we have an example where we caught 20 butterflies. In the second sample we caught 22, and 9 of them were from the original 20 that were marked. So that would mean we had 48.89, but we have to round up to have a whole number. So 49 butterflies. Now you have to consider the ethics here of how you capture them. And you aren't meant to be an expert and know exact suggestions. What you need to know is how you capture them and how you mark them must cause no permanent damage. So don't use anything that's going to be toxic, and it can't increase the chances of predation, and it mustn't interfere with their reproduction either. There are some assumptions with this as well. So the estimate may not actually be very accurate because of these assumptions, because we assume that the population size is constant, meaning there's no birth and death and no migration, which is very unrealistic. We also assume that the animals do redistribute evenly, but that might not be true. They might all huddle together near a food source. So next we have a look at succession, which is the change in an ecological community over time. A primary succession starts with a pioneer species colonising bare rock or sand. And the pioneer species, such as lichen, are adapted to survive in harsh abiotic factors. And through their death and decomposition, they change the abiotic factors to become less harsh, and this forms a thin layer of soil, which we call humus. Mosses and smaller plants are now able to survive in that area, and they further increase the depth and nutrient content of the soil. And this pattern continues, and as the abiotic factors continue to be less harsh, larger plants can survive and they continue to change the environment further. Each new species may change the environment in such a way that it becomes less suitable for the previous species, and therefore those previous species become outcompeted. Changes that organisms produce in the abiotic environment can result in a less hostile environment and increases the biodiversity. The final stage of a succession is known as the climax community, and this is dominated by trees typically. Now, a secondary succession is when the succession is disrupted and the plants are destroyed. Therefore, the succession has to start again, but we don't start again from bare rock because the soil is already there, it's just the plants that have been destroyed. So overall, from succession, as we have that change over time, the species richness will increase, as well as the number of organisms, or in other words, the biodiversity increases over time. As succession occurs, larger plant species and animals will also start to colonize the area. Now, often conservation of habitats involves controlling succession. The destruction of habitats, usually due to human activity, results in a loss of food and a loss of space or habitat for organisms, and it can lead to extinction. So that is why we conserve the habitats, and succession is often managed to do this. So by maintaining an earlier stage in succession and preventing the climax community, a greater variety of habitats are conserved, and therefore we get a greater range of species. There needs to be management, though, between the conflicts between human needs, such as needing the wood for timber, and conservation in order to maintain the sustainability of the resources. So a compromise could be that forests are coppiced to provide timber for fuel, whilst still allowing the trees to survive. Now, this picture isn't actually showing coppicing, this is showing that all the trees have been cut down. Coppicing is when you cut down just part of the tree so they can still then grow. So that is it for topic 7. I hope you found it helpful. If you have, please give this video a thumbs up.